Hello everyone. In this lecture, I'll be covering motor proteins and vesicular transport. I'll begin by discussing phosphatidylinositol and Rab proteins, which they dictate membrane identity. Then I'll talk about some of the Rab effector proteins like chysin or Dynein and how these complexes mediate the microtubule-based transport of vesicles and things like lysosomes or mitochondria. I'll wrap up the lecture by talking about various dysfunctions implicated in neurodegeneration. And if you have any questions or comments, please comment below, and thanks for watching. Before jumping into transport and motor proteins, we need to discuss membranes. Specifically, we need to discuss the component of membranes called phosphatidylinositol. Phosphatidylinositol is the most important membrane phospholipid, I would argue, because the inositol head groups of this phospholipid can be phosphorylated at three different positions. Inositol can be phosphorylated at the 3, 4, and 5' carbons. And these phosphate groups play an enormous role in the cell. Phosphatidylinositol phosphorylation patterns dictate the identity of any given membrane. Let me repeat that. Phosphatidylinositol phosphorylations, the patterns of those phosphorylations dictate the identity of any given membrane. In this graphic, the inositol phosphorylations are abbreviated PI for phosphatidylinositol, followed by phosphorylation sites like 4 or 5, and then P for phosphorylation. So PI3P means phosphatidylinositol phosphorylated at the 3' position. A membrane is a very protein-rich active region of the cell, right? So you have to wonder how those proteins recognize the correct membrane. How does a plasma membrane protein know that it has reached the plasma membrane and not some internal vesicle? And that, of course, is through specific patterns of phosphatidylinositol phosphorylation. The plasma membrane is enriched with PI45P, so inositol that's been phosphorylated at the 4 and 5' positions. PI45P binding domains on proteins include pH domains and ANTH domains. These pH and ANTH domains can actually bind directly to the face of these membranes that have high PI45P content. pH domains, which stands for plextrin homology, are the most common domain used to bind PI45Ps, and they can be found on proteins like Dynein to mediate endocytosis. Dynein is the GTPase that squeezes the neck of an inward budding vesicle during endocytosis. And Dynein localizes to these membranes using its PI45P recognizing pH domain. Some other plasma membrane proteins with pH domains include phospholipase C, protein kinase C, AKT, RAC proteins, and many others. So if you have a peripheral membrane protein localized to the plasma membrane, it's probably doing so using a pH domain to bind to the PI45Ps. Active parts of the plasma membrane are often enriched in PI345P, sometimes called PIP3 in textbooks for simplicity. But you might remember that from introductory biology classes that PIP3 results from receptor activity and various signaling pathways. The inability to dephosphorylate PIP3 back into PIP2 is one of the leading causes of cancer. Something as simple as dephosphorylating PI345P into PI45P is a leading cause of cancer. The phosphatase that mediates this dephosphorylation is called PTEN, PTEN, and it's a potent tumor suppressor because when PTEN is knocked out of the cell, it loses the ability to deactivate PIP3 signaling domains, because it can't dephosphorylate, it can't function as a phosphatase anymore. Secretory vesicles from the Golgi are enriched in PI4P, and in yeast, it's critical for secretion. In mammals, the importance of PI4P in vesicle secretion has yet to be determined. Once these PI4P vesicles reach the plasma membrane, they are actually tethered and anchored by the presence of PI45P binding snare proteins that are on the vesicles. These vesicles contain snare proteins, and these snare proteins bind to the PI45P. This mechanism prevents secretory vesicles from fusing with any old membrane they encounter, and instead they only fuse with PI45P enriched plasma membranes. Because of the snare proteins that mediate fusion are recruited by these PI45Ps. Endocytosis is another process that is highly regulated by PIs. Specifically, we see something called Rab conversion, which we'll discuss in the next slide, but the basic idea is that during endocytosis, the membrane PI45Ps are cleaved off by a phosphatase, and this recruits Rab5. Rab5 recruits a PI3K, or phosphatidylinositol 3 kinase, to produce phosphatidylinositol 3 phosphate. And PI3P recruits another Rab protein called Rab7. So these PI3Ps on endocytic vesicles recruit proteins with 5V domains or PX domains, FYVE domains. PI3Ps recruit 5E and PX domain containing proteins. One important example of this that we're going to discuss in a little bit is the protrudin and the FICO1 proteins. Protrudin is an ER membrane protein that binds to late endosomes by these PI3Ps using a 5E domain. And when it does this, it transfers chysin to FICO1, which is found on the endosome. And FICO1 functions as an adapter to chysin. So I have a slide on this later, so I won't go into more detail here. But the basic idea is that PI3P is recognized by specific domains, such as the 5V, FYVE domain. We'll see why that's important soon. Late endosomes are marked by the appearance of PI5P in addition to PI3P. In other words, the formation of PI35P. And this marks the process of endocytic maturation. And this is done through a protein called PIK5E, PIKFYVE, PIK5E, which, as it sounds, is a phosphatidylinositol 5 kinase that localizes to PI3P using a 5V domain. So it uses the PI3P binding domain 5V to localize to the appropriate membrane, and then it phosphorylates PI5P. In knocking down PIK5E and thus preventing PI35P formation, interferes with endosome trafficking and prevents autolysosomal formation in flies, at least. So the maturation of endosomes towards a degradative fate relies on PIK5E's placement of these PI35Ps. And this is known as a PI3K independent mechanism of autolysosomal formation, which I don't think we get into that here, but don't worry about that. And lastly, we have mitochondria and peroxisomes enriched in PI45P, but currently the research is pretty underdeveloped on that area, and the importance of those phospholipids are disputed. So I'll save that for a future slide. So why am I talking about phosphatidylinositol so much? I said they mediate membrane identity, but how? Well, some proteins bind to these phospholipids, thus localizing them to the membrane. But one thing I didn't tell you is that these interactions are also very weak and transient. So the binding of a PX or 5V domain to PI3P is very weak, or the binding of a pH domain to PI45P is weak and it's transient. In order for a protein to bind one of these membranes and really stick to it, they require another protein. And that other protein is of the Rab class of proteins. The recruitment of any given protein to its membrane often requires two things: the proper phosphatidylinositol phosphorylation pattern and the proper Rab protein. This provides a kind of coincidence detection whereby both conditions need to be satisfied in order for a protein to localize to their respective membrane. And in this way, Rab proteins are kind of thought of as dictating membrane identity. This is how they work generally. So a Rab protein, being a GTPase, is inactive in the cytosol when it's bound to GDP. However, a GEF, a guanine exchange factor, will come around and give the Rab protein its GTP, and this allows it to embed itself into the target membrane using two C-terminal lipid groups called prenyl groups. Technically, GTP-bound Rab proteins actually bind to an escort protein that does this, but the idea here is that Rab GEFs, GEFs, trigger Rab's localization to its respective membrane. Then, in concert with phosphatidylinositol, it will recruit effector proteins to that membrane. Rab proteins can then be dissociated and turned off by GAPs, GAPs, guanine activating proteins. And these hydrolyze the GTP into GDP, and this prevents its interaction with its effector proteins. So Rab GEFs activate Rab, allowing it to embed itself in the membrane, where and once it embeds itself in the GTP bound state, it can recruit effector proteins. Rab GAPs, guanine activating proteins, hydrolyze Rab's GTP into GDP, inactivating it and preventing interaction with effector proteins. Now let's look at how Rab proteins and phosphatidylinositols coordinate together to mediate membrane dynamics. At our plasma membrane, remember we have these local PI35P enriched microdomains that recruit proteins with pH domains. And this includes most notably the proteins from the endocytic machinery, like Dynein and AP2. So PI35P enriched microdomains recruit endocytic machinery, and following endocytosis, various phosphatases are recruited, and some of the most well-known endosome phosphatases are SHP2 and synaptogenin. These phosphatases remove all the phosphorylations from inositol. This process is important for synaptic vesicle recycling, as you may have hinted from the name of synaptogenin, the phosphatase. Membranes deficient in phosphorylated PIs are substrates for Rab5. So Rab5 localizes to these naked endosomes where it recruits a PI3K, simply called VPS34. VPS34 is thus a Rab5 effector protein. VPS35 begins phosphorylating phosphatidylinositols at the 3' position, and these PI3Ps then recruit another Rab protein called Rab7. And Rab7's effectors are distinctly different from Rab5 effectors. In fact, this process of turning Rab5 into Rab7 is very important, and it's called Rab conversion. Rab5 positive vesicles are actually called early endosomes, and late endosomes are Rab7 positive. So these Rab proteins are actually used in the definition of what we call an early or a late endosome. So our definition of what makes an endosome early or late is dependent on either it being Rab5 positive or Rab7 positive. In more specific terms, what happens is Rab5 recruits VPS34, which begins placing PI3Ps. PI3Ps recruit SAND1. SAND1 displaces the Rab5 GEF guanine exchange factor called RabX5. This effectively is displacing the protein responsible for activating Rab5. SAND1 replaces the Rab5 GEF with the HOPS complex, and the HOPS complex is a Rab7 GEF. So HOPS gives Rab7 GTP, allowing it to insert itself into the membrane. This is another look at early to late endosome Rab conversion. So we have Rab5 being activated by RabX5 GEF at PI deficient membranes. Rab5 recruits VPS34, which is not shown here, and VPS34 begins producing PI3P at the membrane, which recruits SAND1. SAND1 replaces RabX5 with the HOPS complex. This causes Rab5 to dissociate when it loses its GTP, and Rab5 is then instead it's activated by the HOPS complex, which gives it GTP. Rab7 then goes on to recruit PIC5E and other proteins involved in generating multivesicular bodies or late endosomes. Remember, PIC5E was the PI3P binding PI5K. So PIC5E binds to the PI3Ps using its 5V domain, and then it begins adding phosphorylations at the 5' positions to generate PI35P. Okay, so this whole time I've been talking about Rab proteins and phosphatidylinositols, but I haven't actually discussed how they mediate vesicular transport, the topic of the lecture. Well, I'm trying to tell a story, and I'm kind of building it up to transport proteins, because really it begins with the phospholipids, which recruit proteins that recruit Rab proteins. And once we have Rab proteins, we have Rab protein effectors that mediate transport. So the general hierarchy is phospholipids, Rab proteins, Rab effectors, and then something called cargo adapters, and then finally our motor proteins. So there's a lot going on in this picture, but it's kind of a summary of where we're going to be going. The basics are that chysin proteins mediate antegrade plus end directed transport, and dynein mediates retrograde minus end directed transport. There's many isoforms of chysin. I think there's 39, but most important for our discussion will be the KIF5 gene coding for the chysin 1 protein. Mutations in chysin 1 are known to cause a motor neuron disease called Charcot-Marie-Tooth. And besides direct cause and effect mutations, many transport-associated proteins have been intricately linked to neurodegenerative diseases simply by the nature of the structure of a neuron. The extraordinarily high energy consumption and complexity of distal synapses and dendrites demands efficient transport and communication with the cell nucleus. And so it's not surprising that the loss of chysin or dynein by experimental knockdown or by some other mechanism is highly toxic to neurons. In fact, a single copy of chysin 1 is haploinsufficient, meaning neurons are so dependent on chysin that they need both copies in order to survive. So given the importance of chysin, it's no wonder that researchers have been probing its possible dysfunction in many neurodegenerative diseases. So let's take a look at some of the worked out mechanisms of chysin-directed transport. Here are a couple of important examples of vesicular transport. We're going to start with the top left. This is a depiction of a synaptic vesicle transport. And I know it's a synaptic vesicle because it contains Rab3. Rab3 is the universal marker of synaptic vesicles, and this chysin homolog, KIF1 alpha, is an isoform highly expressed in the brain and is known to transport synaptic vesicles. This KIF1 alpha can actually link up directly with these vesicles using its pH domain. So we know it's PI3P enriched. In addition, PI3-4 enriched. In addition, KIF1 alpha binds to the Rab3 effector protein called DEN-MAD, DENN-MAD. So synaptic vesicles are Rab3 positive with phosphorylated PIs. This recruits the KIF1 alpha kinesin protein. Then the DEN-MAD is our adapter that bridges our KIF1 alpha to Rab3 positive synaptic vesicles. And mutations in KIF1 alpha are known to cause mental retardation and spastic paraplegia. So we know this is an important process. Next is an example of TRKB positive vesicle transport. TRKB is the growth receptor for BDNF, brain-derived neurotrophic factor, and it can be tethered to kinesin 1. The gene name, again, is KIF5, but it's called kinesin 1, and it's tethered to kinesin 1 by this adapter called CRMP2. TRKB can also be tethered to kinesin 1 via GIP3. GIP3 or GIP, this class of proteins are called J and K interacting proteins. They are a common class of kinesin-associated adapter proteins. In this graphic, GIP proteins are bridging APP, or amyloid precursor protein, and also ApoE2, the receptor for apolipoproteins. In the top right is another transport mechanism for synaptic vesicles. This time it's a protein called syntabulin. Syntabulin is functioning as an adapter between syntaxin positive synaptic vesicles and kinesin. Glutamate receptor positive vesicles are transported by KIF5 via the GRIP1 adapter protein. GRIP1 is an abundant protein found in the postsynaptic densities, and it's thus not only responsible for mediating glutamate signaling at synapses, but it's also important for transporting glutamate receptors to the actual synapse. And for retrograde transport, we can see that Huntingtin is acting as an adapter for vesicles, and that's very intriguing. We're going to discuss that in a later slide. And inhibitory glycine receptors also being retrogradely trafficked by Dynein. Anyways, the point of this slide is to kind of introduce you and to show you that there are dozens of adapter proteins and transport protein homologs that mediate vesicle transport. Many of them overlap. Such a mutation in one adapter isn't necessarily game over for the organism. There are often multiple adapters for any given kinesin homolog. And in the next couple of slides, we're going to focus on more specific examples of anterograde transport. Lysosomes/late endosomes are dynamically shuttled between the cell periphery and the perinuclear region of the cell. While at the cell periphery, these lysosomes are perfectly situated to degrade and recycle various receptors. And this is what happens during basal normal conditions. However, during starvation, lysosomes are moved towards the perinuclear region where they meet up and they fuse with autolysosomes or autophagosomes. So in research into starvation, a big question is how lysosomes and autophagosomes, how they are transported and how that transport is coordinated by nutrient levels. Specifically, how do nutrients cause lysosomes to move from a state of starvation around the nucleus back to the cell periphery during normal conditions? The answer to this question lies in the regulator complex, which is able to sense amino acid levels through the SLC38A9 amino acid transporter. A regulator is also regulated by amino acid binding proteins, cestrin and castor. They're not shown here. Regardless, when the regulator complex senses amino acids, it relieves its inhibition on BORC, B-O-R-C. And BORC is a multi-subunit complex that through the RL8 can link up with kinesin proteins. So amino acids cause regulator to relieve its inhibition of BORC. allowing it to link the lysosome with kinesin through the RL8 adapter. And this allows it to be transported to the cell periphery. What this means is that if you have a culture of starved, nutrient-deprived cells, their lysosomes will be located around the nucleus. And on a molecular level, what is happening is that regulator is inhibiting the Bork complex, and it's preventing it from mediating antegrade plus end directed movement. When you add these cells to a nutrient-rich broth, SLC will stop inhibiting regulator, and this allows the Bork complex to link up with kinesin through its RL8 adapter. And so this is one mechanism of antegrade lysosomal transport that's been worked out. And in the next slide, we're going to examine another model that's also been observed. In this pathway, teased out by a research team led by Camilla Rayborg, they have elucidated the reason why VPS34 activation is necessary for lysosomal peripheral localization. So the previous research showed that amino acids could directly bind and activate VPS34, which was critical for the relocalization of lysosomes to the periphery. And we talked about VPS34 as being a PI3K kinase important for converting Rab5 into Rab7 vesicles. VPS34 activation led to PI3P production at the lysosomal membrane, and these PI3Ps can be bound by 5V domains. In particular, these VPS34 produced PI3Ps are bound by protrudin and FICO1. Both of these proteins do so using a 5V domain. So protrudin and FICO1 bind to these lysosomes using a 5V domain. And protrudin is an ER membrane-bound protein that transfers kinesin 5 to FICO1. And FICO1 is actually a well-known Rab7 effector that functions as an adapter to kinesin. So the basic pathway is VPS34 activation by amino acids. This causes PI3Ps on the lysosome to recruit FICO1 and protrudin. Protrudin transfers kinesin to FICO1, and once FICO1 is functioning as an adapter between kinesin and the lysosome, it's able to mediate antegrade transport. And note that FICO1 also binds Rab7, and this is because, remember, phosphotidylinositols only bind transiently to protein domains. So FICO1 can link up both with lysosomes using both its 5V domain and by functioning as a Rab7 effector. And also note over here on the right that the Bork complex in the RL8 adapter do get mentioned. It's not exactly understood why there's two different mechanisms, but they're both equally viable. In this slide, we're going to discuss how mitochondria are antegradally transported, such as they would be transported perhaps down the length of an axon to a synapse. And unlike lysosomes and other vesicles, mitochondrial transport does not seem to be influenced by Rab proteins or by phosphotidylinositol composition. Instead, mitochondria are tethered to kinesin through these miro and TRAC proteins or by synthabulin. Miro is a mitochondrial outer membrane protein that binds to kinesin through the adapter protein TRAC, T-R-A-K. Early research into this topic found that both Miro and TRAC knockouts produced identical phenotypes where mitochondria were largely unable to move, and instead they kind of packed around the nucleus. And they were shown to co-immunoprecipitate and interact with each other, and indeed they form a complex with each other, and they link up with chysin to mediate antegrade transport. In terms of regulation, Miro contains two EF hand domains. EF hand domains bind calcium ions. This has led to this interesting theory of activity-dependent docking of mitochondria. Because when Miro binds to calcium, it actually drops chysin. When Miro binds to calcium, it drops chysin. And when are calcium ions upregulated? Well, in a dendrite, calcium is upregulated following stimulation by a synapse. So the idea is that mitochondria are ferried up and down dendrites, and if a mitochondrion happens to be passing by a dendrite that is active, that calcium flux will actually bind to Miro and cause it to unload the mitochondria. So by this mechanism, active dendritic spines are more likely to be innervated with ATP because they are essentially signaling to passing by mitochondria to stop and help out. And the binding of calcium to Miro is what triggers the detachment from chysin and the microtubule. Another protein that has been shown to negatively regulate mitochondrial transport is syntaphin. Syntaphin, or abbreviated SNPH, has been shown to be required for activity-dependent mitochondrial immobilization in what's called the engine switch and brake model. Researchers Chen and Shang discovered that SNPH, or syntaphin protein, to bind directly with the ATPase motor domain of chysin and prevent it from transporting mitochondria. And this SNPH protein appears to also be calcium dependent. So the current theory is that when Miro binds to calcium, it partially detaches from chysin. And this actually opens up the binding domain for SNPH to more stably immobilize the mitochondria for longer periods of time. And this model is actually supplementary to the Miro track model as well, because SNPH localizes only to axons and not dendrites. So in dendrites, the Miro track chysin complexes sense postsynaptic potentials by calcium flux, which causes the complex to drop the mitochondria off at active dendrites. In the axons, something similar happens, but when the Miro track complex binds calcium, the SNPH protein actually jumps into its place, and it more permanently immobilizes and docks the mitochondria. And these findings have actually generated a lot of interest in neurodegenerative diseases because knocking out SNPH generates hypermotile mitochondria. And a common theme in many neurodegenerative diseases are immobile and dysfunctional mitochondria that struggle to reach synapses or dendritic spines. And this is especially a problem in motor neuron disease because the length of those axons makes transporting mitochondria makes it uniquely problematic. So perhaps by knocking out or downregulating the SNPH gene, we could possibly increase the motility of mitochondria and get those synapses supplied and innervated with ATP. Syntabulin is another important KIF5 adapter, but it's primarily localized to synapses, and more specifically, synaptic vesicles. Syntabulin is thought to tether synaptic vesicles by binding its syntaxin snare protein on the synaptic vesicle and then linking it up with chysin. In general, syntabulin is important for the formation of the presynaptic active zone, where synapses are moved into position for vesicle fusion and synaptic signaling. But in order to actually reach the synapses, vesicles are often transported by this Rab10 GIP1 kinesin complex. So most synaptic vesicles are generated simply by endocytosis, but to resupply the membrane and other essential proteins, they are usually transported from the cell body by Rab10 positive vesicles, which link to kinesin through the GIP1 protein adapter. However, as noted in the previous slide, many synaptically localized receptors like TRKB can function as kinesin adapters themselves. And also Rab3 is also known to transport synaptic vesicles. So there's multiple mechanisms to move synaptic vesicles down the length of the axon. And defining exactly what a synaptic vesicle is kind of murky. In some cases, vesicular transport actually isn't even relevant because synapses are known to contain many of the mRNA and ribosomes that they need to function, as well as the axoplasmic reticulum needed to sort those proteins. So the question is, how does mRNA get to the synapse? And we'll discuss that in a coming slide. Before moving to RNA granule transport, I wanted to throw this figure in. It's a nice summary of kinesin transport and shows many of the players we've discussed so far. So synaptic vesicles are known to be transported by many different kinesins, probably depending on the specific neuron, the cell type, subcellular localization, developmental stage, and other things like that. Generally, synaptic vesicles are transported by Rab3 positive vesicles linked to KIF1 by the DEN-MAD complex. But as noted in the previous slide, Rab10, GIP1, KIF5 complexes and syntabulin and KIF5 are known to transport synaptic vesicles as well. Below each of these is a list of various kinases. Down here, these are just a list of various kinases that are known to regulate that transport. I'll just point out GSK3 beta. We're going to briefly talk about GSK3 beta when we talk about neurodegeneration because it seems to be upregulated, as does J and K. GSK3 beta, J and K, and CDK5 are all stress-associated kinases. RNA is typically transported in large tangled granules of mRNA. These RNA granules are nucleated by various RNA binding proteins like PURA or pure alpha, and also pure beta, PUR beta, and also some other proteins like stauffin or FMRP. FMRP is particularly interesting because an expanded trinucleotide repeat mutation in one of its coding domains causes a common form of mental retardation. FMRP is thought to not only bind mRNA, but also to function as an adapter to kinesin for transport of these RNA granules to dendritic spines. And the trinucleotide expansion mutation that causes Fragile X syndrome results from an unstable FMRP mRNA transcript that doesn't get translated. And without FMRP, there is widespread mislocalization of normally synaptic mRNA, and this leads to severe abnormalities in brain development. A recent paper by Martin et al., just this year, 2018, they discovered dendritic spine activity to activate sumoylation. It's hard to say that, sumoylation of FMRP. And this causes it to drop its cargo off. So kind of like how calcium flux binds miro and causes it to drop off mitochondria, activity-dependent sumoylation causes FMRP to drop off its RNA granules at active dendrites. Another mechanism of mRNA transport involves the delivery of beta-actin mRNA to actively migrating axon tips by ZBP1. ZBP1, like FMRP, is an mRNA binding protein that can link up with chysin, but it appears to have an affinity for beta-actin mRNA. In general, the field of mRNA transport in neurons is severely underdeveloped. A recent high-throughput analysis of mRNA granules in neurons identified 37 proteins associated with those granules. And teasing out what each protein is doing is incredibly difficult because some are involved in covering, exons. Some might be covering introns. Some might be involved in regulating translation, and some might be just holding it together. And some might be functioning as an adapter to chysin, which is what I'm trying to tease out in this video. But basically, the FMRP, known to as Fragile X, is a very likely candidate for mRNA granule transport. So far, we've mostly been talking about anterograde transport. Now let's transition into talking about dynein, the retrograde microtubule-based motor protein. I don't want to get too much into the specific structure of dynein because, well, it's extremely complicated. And honestly, it's kind of an ugly, strange-looking complex that you wouldn't think was a motor protein. But all you need to know is that if dynein is active, it transports cargo retrogradely towards the cell nucleus. And that is essentially the summary. However, in order to transport cargo retrogradely, it needs to be at the plus end of a microtubule, right? It's kind of so obvious you don't even think about it, but in order for dynein to move something backwards, it needs to get to the end itself. And the plus end localization of dynein is actually accomplished by chysin. So our anterograde motor protein needs to first move our retrograde motor protein to the end of the microtubules, so it can transport things backwards. And there's evidence that there's also evidence that a protein called APC can bind and transport dynein mRNA to the plus end of microtubules, where it can be actually synthesized locally. Regardless, once our dynein is moved to the plus end of the microtubule, it's regulated by two important proteins, Lys1 and Nude, also called NDE1. Lys1 and Nude form a complex that regulates dynein's processivity. Basically, it modulates its speed. And the complex does this through complicated interactions with transient conformations during dynein's power stroke. But the important idea is that Lys1 and Nude are very critical subunits of dynein that generally increase the processivity of the dynein motor. It's believed that NDE or Nude1 inhibits dynein power strokes and basically waits for Lys1 to be recruited before becoming functional, since NDE1 alone actually inhibits dynein. Loss of function mutations in Lys1 causes the loss of brain sulci and gyri and results in a smooth brain phenotype, which is accompanied with mental retardation. This phenotype is likely the result of aberrant neuronal migration due to dynein defects. In vitro research would suggest that Lys1 knockout would drastically interfere with dynein's processivity because it would be constitutively inhibited by NDE1. Because NDE1 inhibits dynein until Lys1 is recruited. Another critical regulator of dynein is the famous dynactin complex. Dynactin is a crazy complex with like 20 different subunits. It's required for plus end localization of dynein, and it's required for dynein function in general. Dynactin is thought of as the dynein receptor for retrograde transport. The main subunits of dynactin are ARP1, ARP1, and P150 glued. Even though dynactin is a subunit of dynein, the P150 glued subunit can actually bind to microtubules itself. And through this mechanism, dynein actually has two different connections to the microtubule. Dynactin, or the P150 glued, kind of tethers it to the microtubule so it doesn't diffuse away. And the dynein itself is what's providing the motor force to move it down the length of the axon. Dynactin is also an adapter to adapters. Dynactin is an adapter to adapters. It's kind of confusing. BICD2. BICD2 is a common and well-studied adapter that bridges dynein and dynactin complexes with Rab6 positive vesicles. And it's a BICD is uniquely important because its mutation causes a common congenital spinal muscular atrophy disease. In the next couple of slides, we're going to look at some more specific examples of dynein transport. In an earlier slide, we talked about how lysosomes and late endosomes are transported anterogradely towards the cell periphery. Remember, anterograde transport of lysosomes occurs through the BORC and RL8 adapters that link up with kinesin. Or in some other cases, Rab7 links up with kinesin through the FICO1 adapter, which is shown in this image. And this process occurs during nutrient flux, when nutrients are abundant. So what about during starvation? What process moves or drives lysosomes to the perinuclear region during starvation? Well, it has to involve dynein, right? Because we'd be moving lysosomes backwards towards the cell nucleus. And lysosomes are identified by the presence of Rab7, which itself, remember, was recruited by the PI3P on the lysosomal membrane. It's also a Rab7 GEF. The critical adapter for retrograde lysosomal transport is this RILP protein, RILP, which stands for Rab7 interacting lysosomal protein. So the anterograde lysosomal transport adapter, so moving it forward to the cell periphery, was that FICO1 over here, and the retrograde lysosomal adapter for dynein is RILP. Although not shown in this graphic, the Huntington protein that causes Huntington's disease is also an important mediator of retrograde lysosomal transport. In fact, we're going to discuss this pretty soon, but Huntington is important for actually many forms of retrograde transport. The question now is, how does this complex, this RILP Rab7 dynein complex, how does it respond to starvation to induce retrograde transport? Well, no one actually knows exactly. It probably has something to do with PI3P, which makes a lot of sense because autophagosomes and late endosome formation, these membranes are enriched in PI3P during formation. This is from kinases like VPS34. Starvation, if you've seen my autophagy lecture, you know that starvation activates ULK1, which phosphorylates Beclin-1. Remember, Beclin-1 and VPS34 are both a part of the autophagic initiation complex. So starvation activates ULK1. ULK1 activates Beclin-1 VPS34, which produces PI3P at the autophagic membranes. And this PI3P recruits Rab7, and Rab7 recruits RILP, which induces the linking of the lysosome with dynein for retrograde transport. But it's still a mechanism that needs to be worked out. But the basic idea of this slide is that Rab7 can direct plus indirected movement when it's transferred, or when something like protrudin transfers FICO1 to Rab7. And retrograde transport can occur when RILP is functioning as the Rab7 adapter, or the Rab7 effector, because RILP is an adapter to Dynein. Another very cool and very important function of Dynein is the retrograde transport of signaling endosomes. So typically when we think of receptor signaling, we think of the plasma membrane relaying a signal towards the nucleus. But did you know that oftentimes the binding of a ligand to a receptor actually induces endocytosis, followed by retrograde transport of that vesicle? So the ligand binds the receptor, this induces endocytosis, and then that active receptor is moved towards the nucleus. And this amplifies the signal by moving the source of the signal right next to the nucleus for more robust gene expression. In neurons, the most studied type of signaling endosomes results from TRKB receptor binding its ligand BDNF. And this induces receptor-mediated endocytosis. And BDNF, brain-derived neurotrophic factor, is, as you might recognize, a potent pro-survival cytokine that inhibits apoptosis. The endocytosis of TRKB BDNF complexes requires the typical clathrin-mediated endocytosis machinery like AP2 and Dynein. And then this complex is, in some way, it's linked up with dynein through dynactin. And interestingly, there's a lot of research that suggests that the Huntington protein that causes Huntington's disease is an adapter for dynactin TRKB interactions. Furthermore, HAP1, or Huntington-associated protein 1, is required for retrograde transport of TRKB positive vesicles, although it's not shown in this graphic specifically. So not only is Huntington implicated in BDNF positive vesicle transport, but a protein that is commonly associated with Huntington is known to be required for TRKB transport. So is this process important? Is this actually an important process for a neuron? Well, studies have shown that when retrograde TRKB BDNF endosome transport is blocked, perhaps by knocking out HAP1 or specifically blocking clathrin-mediated endocytosis, there's actually a massive loss in cell viability. Preventing TRKB BDNF endocytosis almost completely blocks the benefit of BDNF to neurons. So let me say that again. It's really amazing. Preventing TRKB BDNF endocytosis almost completely blocks the benefit of BDNF to neurons. In other words, the binding of BDNF to TRKB, that's only a small part of the process because most the neurotrophic effect comes from the retrograde transport of the endocytosed vesicle. And that means dynein, dynactin, Huntington, HAP1, these are all very important for BDNF signaling. And here on the right, these are all the pro-survival signaling pathways are relayed from these mobile signaling platforms. And the closer it is to the nucleus, the more pronounced the pro-survival gene expression will be. This figure provides a nice summary of retrograde transport. Our Miro tract complex on mitochondria can link up with dynein through GIP1. It's a common motor protein adapter that we were discussing earlier. Our Rab7 positive lysosomes and autophagosomes can link up with dynein through RILP. But in this figure, the authors are referencing research suggesting Huntington, again, is involved in retrograde transport of lysosomes. Downregulation of Huntington causes peripheral accumulation of lysosomes. so lysosomes near the plasma membrane. Interestingly, these Huntington deficient cells still have lysosomes attached to microtubules, but they're just unable to link up with dynein, suggesting Huntington may be involved in retrograde transport, in addition to our conventional RILP Rab7 mechanism. Our signaling endosome, TRKB BDNF, induces clathrin-mediated endocytosis and retrograde transport through the action of HAP1 and Huntington. And lastly, we commonly see retrograde transport of lipid droplets, which basically serve as reservoirs of highly reduced carbon in the form of fatty acids. And Huntington, again, has been implicated in retrograde transport of lipid droplets. One type of regulation that appears to be worth mentioning is GSK3 beta. Again, GSK3 beta can phosphorylate dynein, and this GSK3 beta, which is a very potent stress signaling kinase, it actually functions as a negative regulator of dynein transport and also kinasin transport. And since this kinase is upregulated in many neurodegenerative diseases, it's certainly plausible that it underlies many of their associated defects in transport that we're going to talk about in the next slide. So let's look at some of the more specifically at some of the dysfunctions, mutations, and various pathological processes associated with our molecular motors, kinasin and dynein. The top two figures are illustrating some of the pathological consequences of mutations linked to Alzheimer's disease. PS1 or presenilin-1 is an enzyme that functions to cleave amyloid beta precursor protein, and its mutation results in increased amyloid beta production. And this results in the activation of stress signaling pathways like our GSK3 beta. GSK3 beta, again, is known to phosphorylate and inactivate kinasin and dynein-mediated transport. Thus, amyloid beta accumulation due to PS1 mutations or other amyloid beta-associated mutations will upregulate GSK3 beta stress signaling, and this inhibits transport. Mutations in tau are also intricately linked to Alzheimer's disease, and tau is a microtubule stabilizing protein, and so it probably influences microtubule-based transport. Microtubule post-translational modifications can also influence the ability of transport proteins to bind and move down the microtubules. Specifically, microtubule acetylation at lysine 40, placed by alpha-tubulin acetyltransferase, also called alpha-TAT for short. Alpha-TAT acetylates at lysine 40 of tubulin, and this greatly increases microtubule stability. Recent studies have shown that lysine 40 acetylation, it doesn't affect kinasin motility, but it does promote the stability of a microtubule, and microtubules are obviously required for kinasin and dynein transport. Lysine 40 acetylation is also required for autophagisome fusion with lysosomes, although I probably won't get into that. Anyways, lysine 40 acetylation is downregulated by the presence of mutant Huntington by an unknown mechanism, and that probably has implications for microtubule-based transport. And lastly, as we already discussed, Huntington can interfere with BDNF signaling endosomes. It does this by interacting with HAP1, Huntington associated protein 1, which is, again, required for BDNF vesicle transport. In addition, Huntington upregulates JNK3 stress signaling, which, like GSK3 beta, is known to phosphorylate and deactivate kinasin motor proteins. Motor neuron diseases are also linked to transport defects. Mutations in protrudin, which you remember is involved in transferring kinasin to FICO1 for antegrade lysosomal transport. Mutations in protrudin cause hereditary spastic paraplegia, marked by the degeneration of lower motor neurons. Mutations in spastin, a microtubule severing protein, causes excessive bundling of microtubules and transport defects, ultimately causing spastic paraplegia, just like protrudin. And there are a couple of other HSP mutations, and most of them are linked to endosome trafficking. Mutations directly linked with motor neurons also cause neurodegeneration. For example, mutations in dynein and kynsin cause Charcot-Marie tooth, which is a lower motor neuron disease, while other mutations in kynsin have also been shown to cause hereditary spastic paraplegia. So transport mechanisms are definitely an important avenue of research in neurodegenerative diseases. Axotomy, or axon severing, is another process that relies heavily on microtubule-based transport. Prevailing models suggest that following axon membrane rupture, a massive calcium flux activates a protein called DLK. DLK is a kinase upstream of the stress signaling JNK3 protein. And DLK not only phosphorylates and activates JNK3 following calcium flux, but it also facilitates its dynein-mediated retrograde transport to the nucleus in order to affect gene expression. DLK phosphorylation of JNK is believed to increase its affinity for the GIP3 dynein adapter protein, allowing it to be transported towards the nucleus. The inability to transport JNK3 dampens axonal injury-induced transcription, and the inability to transport JNK3 actually favors entrance into apoptosis rather than regeneration. It's likely that this mechanism of JNK3 signal amplification by DLK is also involved in other pathological conditions and not just axotomy. But regardless, this JNK3 GIP3 dynein complex appears to be formed following calcium-dependent DLK activation, and it's ultimately very important for axon regeneration. Another mechanism of injury-induced retrograde signaling occurs through the local translation of importin mRNA. Imp beta 1, or important beta, is traditionally known to shuttle proteins containing a nuclear localization signal, or NLS, into the nucleus through the nuclear pores. Interestingly, important beta mRNA is localized to axons and synapses by a 3' motif that, when deleted, actually causes embryonic lethality. So this 3' motif on important beta localizes its mRNA to axons, and its translation appears to be calcium dependent, although the mechanism isn't understood. Regardless, calcium flux enters, or calcium flux either from close association with NMDARs or by perhaps membrane rupture causes important mRNA to be translated. Important beta then binds to nuclear localization signals, which is a very common domain on many different proteins, and then translocates the protein to the nucleus through dynein. There's evidence, for example, that the neuronal ERK signaling from synapses requires retrograde transport since its capacity for diffusion is very limited. And at least one way of accomplishing this is by using important beta as a dynein adapter after important beta has been translated. STAT3 is another signaling molecule involved in long-term depression, LTD, and it appears to be critically dependent on important beta-mediated retrograde transport. Ultimately, these are very interesting, but they're actually largely underdeveloped ideas. The basics are that important beta mRNA is localized to axons and synapses. It's translated in a calcium-dependent manner, and it links various nuclear localization signal containing proteins to dynein for retrograde transport. And many of these proteins are actually transcription factors that have important implications for cell viability and cell health. So anyways, that's all I have for transport. If you guys have any questions about this topic or have a request for a future video, please comment and let me know. And thanks for watching.